Hi, today we're going to cover the senses. The senses are going to be the last unit that we cover in Biology 141 or Anatomy 1. With this, it's going to be broken up into two different categories. We'll talk about general senses and special senses. Now, the major body senses, like we said, are broken up into two categories. With the general senses, those are going to be the ones with receptors that are widely distributed through the body, including things such as the skin, various organs, and joints. So we have those such as touch, pressure, temperature, and pain. Now with the special senses, these are going to have more of a specialized receptor to them, and they're going to be confined to structures in the head such as the eyes or the ears. So these will be things such as smell, taste, hearing, equilibrium, and vision. Now with the general senses, you're going to have different types of senses that we're going to go over in detail here. And so with these are going to be those with sensory receptors that are going to be widespread and associated within things such as, like we said, skin, muscle, joints, and the viscera. These senses are divided into three different groups. So you have your exteroreceptor senses that are associated with changes in the body surface. They include the sense of touch, pressure, temperature, and pain. Next, you have the interoreceptive sensors, or sometimes referred to as the visceroreceptive senses. These are associated with changes in the viscera. So things like blood pressure, stretching blood vessels, and ingested meals stimulating chemoreceptors in the small intestine, and so on. The third type is going to be called the proprioreceptive senses, and these are going to be associated with changes in muscle and tendons and body positions. This information from these senses are going to be sent to the spinal cord and can help mediate the different reflexes that need to help respond to these stretching. So in with this, we're going to have different receptor types. And the 5 types of sensory receptors are going to be recognized and based on their sensitive and specific stimuli that they're going to come in contact with. So first, starting out, we're going to have the chemoreceptors. The chemoreceptors are going to respond to changes in concentration of chemicals. These receptors are associated with senses of smell and taste that are going to be this type. You're going to have chemoreceptors in places like internal organs that will detect changes in blood concentrations, for example, of oxygen, carbon dioxide, hydrogen ions. They could detect glucose concentrations and other chemicals that you may find in these organs or these areas. The next type of receptor is going to be your pain receptors. These are also called nociceptors. And so with these, they are going to respond to tissue damage. These trigger stimuli that include exposure to excess mechanical, electrical, thermal, or even chemical energy. The third type is going to be your thermoreceptor. These are going to respond to changes in temperature. Next, we have our mechanoreceptors. There's going to be several types, and they respond to mechanical forces by detecting change that often deform the receptor. They include a number of receptors in the skin that respond to physical contact, and several receptors that are going to be in places like the ear that provide information about balance and vibration from sound. You have your proprioreceptors. They're going to respond to changes in tension of muscle and tendons. The baroreceptors, which are going to be called also the pressure receptors, and they're going to be found in certain vessels to detect change in blood pressure. Then you have your stretch receptors that are going to fall in this category also, and these are going to be found in places like the lungs that can determine the degree of stretching or inflation of the lung. The fifth one is going to be the photoreceptors. So these are going to be in the eyes, and they respond to light energy of sufficient intensity. So if, for example, you have a lot of light that's coming in, it's going to cause the contraction of that pupil. And if it's going to be dim light or not a lot of light, you're going to have a dilation of that pupil. Now here we're going to see a chart. In this chart, it's going to tell the type of nerve ending or the type of receptor, and it's going to tell you the function and the sensation. So with free nerve endings, these are going to be mechanoreceptors. The function is to detect change in pressure. Here, that's going to be the sensation of things like touch and pressure. You have your tactile corpuscles. These are in the skin, of course. These are also mechanoreceptors. They're going to detect objects that are moving over the skin. So this would give you touch or texture. It'd tell you if things are smooth or if they're rough. Lamellated corpuscles are going to be mechanoreceptors as well. They detect change in pressure. And so this will be your deep pressure. This gives you vibrations and fullness in the viscera or your organs. You have free nerve endings. These could be thermoreceptors. They're going to detect changes in temperature, so they pick up on heat and cold. You have free nerve endings that are going to be pain receptors. They detect tissue damage and tell you if it's painful. You have free nerve endings that are mechanoreceptors that detect stretching of tissue and tissue spasms, so that will be in visceral pain. Muscle spindles are going to be mechanoreceptors, and they detect changes in muscle length. Golgi tendon organs, these are going to be mechanoreceptors, and they also detect changes that are going to be in muscle tension. Now here you're looking at a diagram of the skin, and it's going to show you several of these different nerve endings. So starting from the top, you have those free nerve endings. Free nerve endings are going to be like fibers that are going to come up. They will stretch, as you can see, just into the upper layer of that epidermis. They don't go very far. Most of the time they do stay in the dermal area, but the top portion of the dermal area. So you can see those epithelial cells that it's pointing at, the bottom one that's going to be the darker pink color. Those are going to be your stratum basal layer. Right up above that, you're getting into the other stratum layers. And you can see how that sensory nerve fiber dives deep and will go through the dermis, through the hypodermis, and make its way through the body. Next, going to picture B, you're going to see that you're going to have your tactile corpuscles, or sometimes referred to as the Meissner's corpuscles. These are your touch receptors that pick up on touch. It's attached to a sensory nerve fiber that then dives deep through the dermis, through the hypodermis, and goes on in and connects with the central nervous system. And the bottom one, C, is going to be your lamellated, or sometimes referred to as the Paxinin corpuscle. That's a pressure receptor, and so that's going to pick up on pressure. It also has connective tissue that's going to be found around that corpuscle, and a sensory nerve fiber that dives deep as well. Looking over to the side for B and C, you can see what they actually look like under a microscopic picture, so if you're looking at it through a slide. Next, we're coming to where we see these sensory nerve fibers that are going to dive deep and wrap themselves around those different muscle fibers. So these type of receptors are going to be your proprioceptors. They are a type of mechanoreceptor that's going to send information to the central nervous system about body position and length and tension of skeletal muscle. Remember that the laminated corpuscle function is going to be a pressure receptor in the joints, while the other main proprioceptors are going to be stretch receptors that are going to be found in these muscle spindles and the Golgi tendon organs. So you can see that here. Those muscle spindles that you can see in the left picture are going to be located throughout skeletal muscle. Each spindle is going to consist of several small modified skeletal muscle fibers. And with these fibers, they're going to be enclosed in a connective tissue sheath. Each intrafusal fiber has near its center of its specialized non-striated region with the end and sensory nerve fiber wrapped around it. And so with the striated portions of the intrafusal fiber, it's going to contract as needed to keep that spindle taut at different muscle lengths. So this is going to be the whole muscle is going to help it stretch and help that muscle spindle also trigger a sensory impulse on this nerve fiber that you see wrapping through it. These sensory fibers will synapse in the spinal cord of the lower motor neurons, leading back to the same muscle. So in this way, that stretching of the muscle spindle is going to trigger impulses that contract that skeletal muscle, which it's going to be part of. And that action is going to be called a stretch reflex. So it opposes the lengthening of that muscle and helps maintain the desired position of the limb despite things such as gravity and other forces that are going to be acting on it. So, for example, the patellar reflex is going to have this stretch reflex. A leg cramp in the middle of the night may be painful, but it's going to make help make you leap out of bed because of that contraction, and it may result in local changes of, let's say, electrolyte levels. That's going to be part of that muscle, an appropriate trigger for this impulse that you see going through the spindle. Now with that Golgi tendon, this is going to be the picture that you're going to see on the right-hand side. These are going to be located in tendons that are going to be close to their attachments of muscle. Each Golgi tendon organ is going to connect to a set of skeletal muscle fibers, and it's innervated by a sensory neuron. So Golgi tendon organs are going to have high thresholds. So they're going to be activated only when muscles are going to be stretched nearly to their limit. So sensory impulses from the produce of that reflex are going to be inhibited further by the contraction of muscles whose tendons that they occupy. So the Golgi tendon organs then are going to be stimulated for a reflex with the effect that it's going to be opposite of that stretch reflex. So this Golgi tendon reflex also helps to maintain things like your posture. It protects muscle attachments from being pulled too loose by excessive tension. And so with this receptor is going to be part of those general senses that are going to help to protect the muscles, basically. Next, we're going to talk about sensation, perception, projection, and adaptation. So with the sensation, this is going to occur when sensory neurons reach a threshold, and it results in action potential that's going to cause the brain to become aware that the sensory event is happening. Perceptions when the brain is going to be able to interpret those sensory signals that are coming to it. For example, you might have the sensation of stepping on a tag, but perception is being able to perceive that that's what you just stepped on. Projection is going to be where the brain is able to project that sensation back to its sensory receptor source. This is going to allow the person to pinpoint the region of stimulation. So in this way, we can perceive that our eyes see that thumbtack that you just stepped on, and it's going to be able to tell that that pain is coming from that thumbtack. Next, we have adaptation. That's going to help us to be able to ignore, let's say, unimportant stimuli that might be there. Because sometimes you have all the stimuli coming in, and you need to be able to tell what's important, what you need to recognize, and basically what you also need to ignore. So with this diagram, we're going to put everything together and see how it is going to receive the information and process that information. So the screen for the recorder doesn't cover all this, so I'm going to have to move the screen as we go through. So we're starting out here at the bottom. We have fine touch, proprioreceptor vibration coming in. Also, you're going to see it come in, and it's going to hit coming in through these nerve fibers through the spinal cord, and it's going to come up to that medulla. From that medulla, it's going to take that fine touch, vibration, and proprioreceptor pathways. They're going to cross the midline in the medulla. They're going to travel up to the thalamus. Here, that sensory pathway synapse in the thalamus is going to pick up on that information. And then we have another synapse that's going to send it further up. So I'm going to try to move the screen from, well, that didn't work, sorry. Go back. Trying to move the screen up. Hold on. That's a little better. So I've got everything in here. So once it goes up to the upper portion, we see those sensations are going to be perceived in the primary somatosensory cortex there. And we can see how it is going to be processed. Now, same thing with the nociception, the temperature and coarse touch. It's going to come in, it's going to travel through. You see how this one, though, is going to travel through a long nerve fiber. It's going to have a synapse at that thalamus. And from the thalamus, it's going to travel up to that sensory cortex. All right, information flow and how it's going to process. So we have sensory receptors. First one we're going to go through with smell. So we have the olfactory receptor as its sensory receptor. It's going to travel through the olfactory nerve fibers to the cerebral cortex. You interpret it's a pleasant smell, and then the smell is going to be perceived as, let's say, an apple. Next, you could have taste. So the taste bud is going to be the receptor cells. It's going to travel through sensory fibers and various cranial nerves to the cerebral cortex. There you get a sweet taste, and then your perception of that taste is going to be interpreted as an apple. Next, you go on to sight. You're going to have rods and cones in the retina that travel up through the optic nerve. The optic nerve takes it to the midbrain and then the cerebral cortex. A small round red object is going to be picked up, and that sight is going to be perceived as an apple. And third, you're going to have last, you're going to have your hearing. So hair cells in the cochlea are going to pick up and send information through the auditory nerve fibers. They travel through the midbrain to the cerebral cortex. You have a crunching sound, and that sound's going to be interpreted as biting into the apple. Pain is going to be an unpleasant sensation that's going to be sent to the brain when tissue is going to be damaged. Tissue damage can be detected by nociceptors. These are your pain receptors on the free nerve fibers. These are going to be located everywhere in the body except for the brain nerve tissue. Nociceptors respond to hydrogen ions, potassium ions, intracellular proteins, histamines, hypoxia, which is going to be lack of oxygen, and high and low temperatures. You can have fast pain, which is going to be A delta fibers. These are going to be myelinated, fast, and they create a sharp pain sensation. Or you can have slow pain, which is going to be found on C fibers. These are going to be unmyelinated fibers that are going to travel with a slow signal and create a dull chronic pain sensation. So in an event that's going to stimulate your pain receptors, this impulse is going to be both fast and slow pain fibers usually. So it's going to result in a dual sensation. Sometimes it's a sharp pricking pain, also followed with a dull aching pain that's going to be more intense and may worsen over time. So chronic pain is going to resist the relief and control, and it can be debilitating. So with these pain impulses, if they originate from the tissues in the head, they can reach the brain and sensory fibers of the 5th, 7th, 9th, and 10th cranial nerves. All other pain impulses are going to be conducted on the sensory fibers of spinal nerves, and they can pass into the spinal cord by the way of the posterior roots of the spinal nerve. So that's what you're looking at here on that left side. So upon reaching the spinal cord, the sensory fibers are going to enter into the gray matter of the posterior horn. There, they're going to synapse with neurons whose axons cross over to the opposite side of the spinal cord at that level, and then they're going to ascend into the anterior and lateral spinothalamic tracts. Here, these pathways are also going to involve one or more interneurons before reaching the long fibers that are going to cross over and then ascend into the brain. The brain. Most of the pain fibers will synapse in the reticular formation, and from there impulses are conducted on fibers of still other neurons to the thalamus, hypothalamus, and cerebral cortex. Fibers of the spinothalamic tracts are going to bring pain and temperature information directly to the thalamus. Once in the cerebral cortex is going to determine the intensity of pain and its source. It's also going to be responsible for motor responses to that pain. So the emotional response to the pain is going to involve that limbic system that you see there. Still, you're going to have other parts of the brain, including areas like gray matter in the midbrain, the pons, the medulla oblongata. That's going to regulate the flow of pain impulses from the spinal cord. So these impulses from the spinal neurons in these areas are going to descend into the lateral funiculi to various levels within the spinal cord. These impulses are going to stimulate the end of certain nerve fibers to release biochemicals that can block pain signals by inhibiting presynaptic nerve fibers in that posterior horn of that spinal cord. Sometimes pain might actually feel like it's coming from a different area. For example, here, pain originating from the heart may feel like it's coming from the skin because sensory impulses from the heart and the skin will follow common neural pathways to the brain. So it makes you think that your arm is hurting when it's actually a portion of your heart that's hurting. Here, you're going to see surface regions to which visceral pain may be referred. So, for example, everything in red will travel through the same neuron pathway to the brain. Everything that you see in yellow will travel through the same pathway. So you could have kidney pain, but it might be picked up on the groin area. It could be picked up on the lateral thighs, on the medial thigh. You could have ureter problems, and it could be picked up on the inner portion of the thigh, the medial section. You could have a small intestine pain, and it'd be picked up anywhere above the umbilical region. You could have liver and gallbladder pain that could be picked up in that midsection that you see there, more closer to the diaphragm and thoracic region, and even picked up on the neck that you see in that region. Pain medications. One that you're probably commonly aware of is going to be your NSAIDs. That stands for non-steroidal anti-inflammatory drugs. These include things like aspirin, ibuprofen. These are going to help to knock out COX-1 and COX-2 enzymes. They're analgesic. These are anti-inflammatory and antipyretic. You have acetaminophen. These most commonly known are by a brand name such as Tylenol. For these, it's an unknown mechanism of action. They're also analgesic and antipyretic, but not anti-inflammatory. So if you have inflammation, you want to use NSAIDs. If you have pain, you can use either one. And so that's what they're going to mean by pyretic. Also is going to help with a fever, because pyrogens cause a fever. You have your opioids. These are going to be things such as morphine, heroin, oxycodone, and hydrocodone. What these do is they're going to block the voltage-gated sodium channels and open potassium channels in order to stop nerves from sending the pain signals. You have anesthetics. These are going to be things such as lidocaine or benzocaine. These are going to block voltage-gated sodium channels. Now that's going to finish us up with our general senses. Next, we're going to head to our special senses. These are going to be senses that have sensory receptors associated with complex sensory organs that are going to be found in the head. The first one is your sense of smell, and we'll go into each of these in detail, but we're just going to list them at first. So you have smell, which is going to be part of your olfactory organs that are going to be found in the nasal cavity. Next, you have taste. These are going to be taste buds, which are going to be found on the tongue. Sight, that's going to be in your eyes. Hearing and equilibrium can be found in the ears. So starting out with the olfactory sense. Olfaction is going to be the name for sense of smell. Now looking here, you're going to see that the route of smell is going to go in through the air that you're inhaling. So it comes up through those nasal concha, travels up to the upper or superior portion, where you're going to see those olfactory bulbs with the olfactory nerve endings that are going to be coming out. They are attached to the olfactory bulb, and it will travel through that olfactory nerve to the brain in order to be interpreted. Notice how those olfactory nerve endings that are coming down into the nasal area are going to come down through that ethmoid bone. And so there's going to be small holes that you'll be able to see there where these nerve endings come down. And then when they go up through that bone, they're going to attach to that olfactory bulb and travel through that olfactory tract. Now looking at it a little bit closer. So here you're going to see olfactory receptor cells, and they have membrane receptor molecules that are going to sense odors here. The receptor cells are going to be similar to those such as the ones that are found in taste, because they're going to be chemoreceptors that are sensitive to chemicals that are dissolved in liquids. Now, two chemical senses are going to function closely together that help to aid in taste and smell, things like that. And notice how they're going to have olfactory cilia that are coming down. And so with these olfactory cilia, notice how they're going to be in that mucus. That mucus is going to be wet, and it's going to require that in order to pick up on those chemicals that are coming in and to be able to tell that sense of smell. So these olfactory organs are going to appear yellowish brown masses. That's what they look like in that epithelium. And they're going to cover the upper parts of the nasal cavity found in that superior nasal concha and a portion of that nasal septum. The olfactory organs are going to contain those olfactory receptor cells that we just talked about, which are going to be bipolar neurons, and they're going to be surrounded by columnar epithelial cells. These neurons are going to have knobs at the distal ends of their dendrites that are going to be covered with that hair-like cilia I just pointed out. Now, the cilia projects into the nasal cavity, and these are going to be sensitive to portions of the sensitive portion of the receptor cells there. And so that actual olfactory receptors are going to be membrane proteins that are going to be associated with the cilia. So each person is going to have about 12 million olfactory receptor cells and about 10 to 20 cilia per cell. So that's going to be a lot, and that's what's going to help you pick up on your smell. So inhaled chemicals that are going to stimulate these olfactory receptor cells are going to be called odorant molecules. So they're going to enter through that nasal cavity, and they're going to dissolve at least partially into those watery fluids that you see that are going to surround the cilia before they can bond to the receptor proteins on the cilia and be detected. So things like foods, flowers, and a lot of other objects and substances are going to release these odorant molecules. Those odorant molecules can bind then to several of the almost 400 types of olfactory membrane receptors that are going to be part of the olfactory receptor cells. This is going to cause a depolarization of those cells and generate action potential if the depolarization reaches a threshold. Now, once those olfactory receptor cells are going to be stimulated, those impulses are going to be conducted along their axons through tiny openings in the cribriform plate of that ethmoid bone that I talked about. So these fibers are going to synapse with neurons that are going to be located in enlargements called the olfactory bulbs that I've also pointed out. This is going to be where they're going to then travel through either side of the crista galli through that ethmoid bone. And so in those olfactory bulbs, the sensory impulses are then going to be processed. And as a result, in addition to impulses, they're going to travel along those olfactory tracts to portions of the limbic system. That's going to be a brain center for memory, emotions, and it's going to be related to behavior. This is why you can become nostalgic over the scent of something from the past that you might remember. Now we're going to head into sense of taste. This is called gustation. Now the sense of smell was olfaction. But now we're going into gustation. So with the sense of taste, taste buds are going to be the special organ of taste. They're going to resemble orange sections and are going to be located on the surface of the tongue associated with tiny elevations called papilla. Taste buds are also going to be scattered on the roof of the mouth, the linings of the cheeks, and they're going to be on the walls of the pharynx. Now, each taste bud is going to include a group of modified epithelial cells called taste cells or gustatory cells. These are going to function as a sensory receptor for this sense. Taste cells are going to have vesicles that contain neurotransmitters. These are going to be interwoven among and wrapped around the taste cell as a network of sensory fibers who's going to end closely in contact with the receptor cell membranes. So each of our 10,000 taste buds is going to house about 50 to anywhere around 150 taste cells. Now these taste buds also will include epithelial supporting cells that help to take care of them as well. So the entire structure of a taste bud is going to be somewhat spherical with an opening and a taste pore on its free surface. You're going to have tiny projections called microvilli. These are going to be referred to as taste hairs. And so they're going to protrude from the outer ends of the taste cell and just through the taste pore. These taste hairs are going to be the sensitive parts of those receptor cells. So the sense of taste is going to be derived from food molecules that are going to bind to specific receptor proteins that are going to be embedded within these taste hairs on the taste cells. So in response to the taste cell, it's going to release a neurotransmitter that's going to alter the membrane polarization on the associated sensory nerve. So here you're going to have a degree of change in polarization, and that's going to be directly proportional to the concentration of stimulating substances. So if you have a sufficient stimulus, it's going to start to trigger an impulse. That's going to be conducted to the brain. So you're going to have a particular taste sensation result from a pattern of specific receptor cells that are going to bind to specific molecules. And so with these molecules, it's going to depend on their shape. So shape is everything when it comes to chemistry. And so these are going to be chemical molecules that have a specific shape. And so sweets will have a specific shape. Salty will have a specific shape. Sour has a specific shape. All of those different flavors have specific shapes, and they fit into specific receptors, which send a signal to the brain that, hey, this is sweet, this is salty, this is bitter, things such as that. Altogether, when talking about taste, there's going to be 5 primary taste sensations. These are going to be sweet, sour, salty, bitter, and umami. Umami is going to be savory. It's kind of a weird name for it, but that's what they call it. So with these, the taste cells for each of these 5 taste sensations are going to be on all areas of the tongue, but they are distributed such that each sensation seems to arise more strongly in particular regions. So here, if you look down at the bottom right-hand corner, you're going to see that sensory impulses from taste receptor cells that you see here in this picture in that anterior 2/3 of the tongue are going to travel on fibers of that facial nerve or facial nerve 7. And these impulses from this receptor in the posterior 1/3 of the tongue and the back of the mouth are going to pass along the glossopharyngeal nerve, which is going to be nerve 9. And impulses from receptors at the base of the tongue and the pharynx are going to travel through the vagus nerve. So you see how they have these nerve fibers in the taste cells. They're going to travel down through those sensory nerve fibers. And then here's where they're going to travel in through the different areas. And so with these, the cranial nerves are going to conduct that impulse into the medulla oblongata. From there, those impulses are going to ascend to the thalamus and then are going to be directed to the gustatory cortex of the cerebrum. This is going to be located in the insula. And so here you can see how it travels up. So sweet, sour, salty, bitter, and umami. And pain perceptions also are going to be sensed here by the tongue and travel up through these different nerves. So both olfactory and gustatory senses are going to use chemoreceptors. So we've referred to these chemicals. For example, the chemical shape determines its taste, or how the chemical dissolves into that mucus in the olfactory senses is going to help to determine the smell. So these chemicals have to be dissolved in a watery solution. Watery mucus secretions in the nasal cavity and in the saliva in the mouth cavity. Olfaction is sensed in the olfactory cortex located in the temporal lobe, where gustation is going to be sensed in the gustatory cortex located in the insula. We're going to stop here. This is going to finish us up for our first part of senses. We're going to pick up with the sense of sight and continue on with hearing for our part 2 video.